Are y'all ready to meet some really cool fungi? The ectomycorrhizal stuff?
Let's do it.
All right. Let me share my screen with you. Oops, wrong glass. Okay. So you may have noticed the reading assignment for this week doesn't exist. Okay, that's on purpose. As a matter of fact, we're not even going to have class on Thursday. I want you to have time to catch up on all your lab work because I'm about to do a big grading binge. Okay. So if you're more interested in ectomycorrhizal fungi after today, I highly recommend this paper right here. It's a review paper in Nature, basically trying to trace back the multiple origins of the ectomycorrhizal lifestyle, and they do a really good job of kind of looking at what goes into being an ectomycorrhizal fungus and how it evolved. Pretty good stuff. Okay. Before we dive right in, any questions about assignments, exam, that kind of stuff? Guess that's a no. All right. So mycorrhizae just means fungus root, okay? And so we've met some mycorrhizal fungi before. Remember, they were kind of buried in the zygomycetes way back in the beginning of class. Those are arbuscular mycorrhizal fungi. Okay. Today our focus is going to be on the ectomycorrhizal, and those are different in that they don't actually penetrate into the plant cells. They're just in that interstitial space between the cells. Okay. And we'll just take a look at their diversity and a bit about their evolution and then see some examples. So this is a mycorrhizal root pictured here. And this is like a very classic, heavily colonized plant root. They get all stubby looking. It's kind of hard to mistake this for anything else when you see it, often kind of with like a light white coating. Okay. So what is this relationship all about? The fungus and the plant. It's mutualistic, but who's getting what? Does anyone have thoughts?
Seems that material is being traded for energy in not too much detail.
Can you be a little more specific?
It seems that we're getting, based on this chart, you got these phosphates and nitrogen that's getting transferred between the fungus. The fungus is doing some manufacturing things to them and then transporting them to the plant. It seems that the polyphosphates are being broken down, and as is fairly typical for endosymbionts, at least, the trade-off is that it's being provided some form of energy by the host.
Yeah. Be careful with that term endosymbiont, though, because these actually aren't outside of the cell at all. But they're pushed up next to the plant cell walls, and they're able to transfer, mostly nitrogen and phosphorus, okay? And then in exchange, they're getting carbon, just simple sugars and stuff from the plant. Because plants are really good at turning air and sunlight into sugar. Okay? Fungus can't do that. It's got to use organic fixed carbon sources. So it's getting sugar from the plant. In return, it's providing phosphorus and nitrogen. Okay. This is a pretty well-studied mutualistic association because so many plants are dependent on it, like all of our forests for the most part. So we've done a lot of kind of like cell bio and molecular work, kind of seeing how this happens. There's still work to be done, but we found transporters in the fungus and in the plant that are like tailor-made, lock and key fit to each other. So there's been some serious coevolution in these groups. Okay. Now this is to kind of get at the point that these are not endosymbionts. Okay, they're not inside the cell, they're actually just in between the plant cells here. So the arbuscular mycorrhiza that we saw before is intracellular, and it actually makes these little arbustles that grow down into the plant cell where it's like exchanging nutrients and materials with them. But for ectomycorrhizal, they're growing on the outside of the roots, and they kind of penetrate in a little bit between the cells like this. And that's where we're getting our transport. Okay. So I said this is mutualistic, and that means they're both, the fungus is giving nitrogen to the plant in exchange for carbon. What else might the plant be getting out of this deal besides nitrogen, phosphorus? Because plants are capable of getting nitrogen and phosphorus on their own.
Is this another example of the fungus taking up a niche that would otherwise be filled by other fungi?
Yeah. So yeah, really good. I mean, imagine their entire root essentially is like coated in this fungus. So pathogens have like a wall of protection around them. Okay. So we see here, this is pathogen accumulation rates, right? Oh, there's my cursor. So pathogen accumulation rate with conspecific seedling effect or conspecific adult effect. So what that's talking about is how many pathogens you get, basically, how to put this, as the plant kind of is growing and maturing. Okay, we have this kind of decline, but at the same time, ectomycorrhizal fungi often will just be called ECM fungi. Okay, those are accumulating at the same rate. So as they're going up, then pathogens are actually going down, and this is without worrying about the nitrogen carbon kind of exchange stuff. So yeah, they're doing a good job of protecting plants from pathogens, and in the meantime they're also trading nitrogen in a nice digestible form for the plant. Okay. So as far as their evolution, of course we thought that they were all one group back in the day, and then started getting better phylogenies, and it turns out no, they're spread out all over the place. Every time you see a star here, that's showing a derived new ectomycorrhizal lifestyle. Okay, so it's many times this has evolved in a lot of different groups of fungi over time. So this particular study was also looking at gene copy number for saprotrophic peroxidases on the left and laccases on the right. What, remind me, is a saprotroph, and then what might a peroxidase or laccase do?
Saprotroph eats dead stuff.
Okay. So what is a blank ACE?
ACEs destroy things. They're an enzyme.
Yeah, so these are enzymes to break down dead material. Okay, so complex material gets broken down, destroyed, made more simple. Saprotrophs in the fungal world are like biochemical masterminds with their ACEs, right? They have all these crazy enzymes that can break down really complex stuff, even like lignin, right? So they're looking at how many of these are retained in ectomycorrhizal groups versus non-ectomycorrhizal groups. Okay, and it looks like in some cases they've really lost a lot of their ability to break down these complex carbon sources. How does that relate to their lifestyle now?
You basically get it from the plants, right? I mean, the plant provides the sugar, plain sugar.
Exactly, yeah. So they're kind of like stuck at this point, being ectomycorrhizal. They used to be able, their ancestors could break down these carbon sources found in the soil, and they could get their own carbon that way. They've gone soft, in essence. They're used to eating like pure sugar now, straight from the plant. And so those genes have been allowed by natural selection to kind of break down because it wasn't damaging to the fungus. So now, obligate biotrophs. Okay, so saprotrophy isn't necessarily a stable way of making a living. If you find a way to get simple sugar, there's no need for you to make these big, expensive enzymes. Okay, now these aren't the same. It's not the same pattern when we look at other genes for like breaking down nitrogen, for example. That's because the plant isn't giving them nitrogen. It's just giving them that sugar. Okay. Roughly, I don't know how good this number is, but I can't find a nice, good, solid number. But what I've seen is about 2% of plant species are ectomycorrhizal. But we still study these things all over the place for a number of reasons. One, they tend to make really nice, pretty, charismatic mushrooms, like that red one with the white spots, the fly agaric. And so they're kind of fun to study. But also because the plants that they do associate with, it's not just 2% of all the individuals on Earth, it's 2% of the species. We're talking about things like the entire taiga belt is ectomycorrhizal dependent. Okay, so we look through here, all the purple are clades that have ectomycorrhizal groups in them. So what do we see here? Pezizales, what's that from? Sorderiales. Anyone remember? Back when we were getting to know the fungi the first month of the class? You've got this one, Sorderiales, growing in a dish. Those are ascomycetes. Shame on you. You should have known that. Okay. And then how about over here? The agaricales, bolitales, russelales. What phylum?
Pseudomycetes?
Yeah, pseudomycetes. So these things are pretty widespread, and they're not necessarily like one species of fungus can only do ectomycorrhizal stuff with one species of tree. Okay, some of them are very host specific. Some of them have pretty broad ranges. What that leads to is kind of a weird phenomenon known as mycorrhizal networks. Okay, so from the picture over here on the right, the top picture, we kind of see the flow of carbon here in yellow, okay, and the flow of water and minerals here in blue. Okay, so like mineralized nitrogen, all that good stuff, or organic nitrogen rather. Okay, what is this actually depicting though? If you follow that yellow arrow,
that flow of sugars?
Mm-hmm. And it's flowing, it's being generated here in these leaves, flowing down to the roots, and then the mycorrhizal fungus that's associated with those roots is getting those sugars, but also this plant. How does that plant get the sugars from this plant?
Is
It's
it
connected to the
because
same
the
system.
fungi are kind of acting as a bridge between the two?
Yeah.
And being able to share the sugars, I guess you could say?
Yes. So this is pretty weird. It's relatively new that we've known about this and had good studies on it, it's past 10 years. If the fungus is interacting with two separate individual plants, the sugars or whatever from this plant can go into that fungal network and actually wind their way into the next plant over. Okay, so we've seen this in little experiments, right, where we've tested this out, where we let the two plants link up mycorrhizal fungi. So we put basically like filter paper that's small enough that no roots can get through, but the tiny little mycelium can. And we've tracked the flow of nutrients from one plant into another through the mycorrhizal network. So this gets to be a bit weird because it's not just sugars that the plant is sending through the mycorrhizal network. We also found that defense compounds are transmitted. Okay. We found small RNA molecules that initiate like cascade pathways that trigger plant defenses are being transmitted from one plant to another, or even poisons. So like the acacia trees that grow down in the tropics, the ones that associate with the ants that attack anything that touches the tree, those ants are brutally painful, by the way. Do not touch an acacia tree. But the ants are out there like chopping down little seedlings all around the trees because they're basically protecting their home. But the acacia tree is also linked up through mycorrhizal networks, and it sends out poison. It doesn't harm the fungus, but gets transmitted through the fungus, and so if anything tries to associate with it, it dies. This is crazy. We've also found plants essentially talking to each other, in essence. Okay. So in an experiment like this one, we can go in and just with a pair of scissors, cut one of the leaves on the plant on the right. And then within an hour, the plant on the left is exhibiting the exact same defense mechanism pathway that the plant that was cut is exhibiting. Okay. So think for a second about what that means for plant defenses. How is that useful in like a real world scenario?
I know if you put it in aspects of like a herd mentality, it protects the whole grove instead of just one or two trees.
Yeah, there was actually a series of pretty cool experiments done using like herbivore, like so insect pests, and they put caterpillars on one plant. They start chewing on it, and that actually sends calcium ion pathways through the plant, and it starts responding like, hey, I'm being damaged. So it starts making compounds kind of fight off the infection. That same pathway gets triggered in the plant next door that's not being eaten. And so suddenly it's less appetizing to the insects. So it's kind of like like herd mentality, I suppose. Like they all start acting like they're being chewed on if one does. And that's connected through the mycorrhizal network.
So could it potentially allow chemicals be transferred through the tree as well to actually help protect the one being chewed on? I guess you could say.
Yeah, and in a sense, that's exactly what's happening.
That's what's happening. Okay.
Yeah, it's not, to my knowledge, it's not like the plant on the right here is actually sending the defense compounds to this plant. It's sending the molecule that triggers the production of the defense compounds.
Okay. Okay,
So,
that makes sense.
yeah, those are smaller molecules, you know, like little small RNA molecules and stuff like that. So they're easier to send through a tiny mycorrhizal network. And in that sense, this plant isn't actually expending much of its own resources to help out its neighbor, right? It's just sending a signal that says, look out, being chewed on. Okay.
So
Oh, sorry.
I'll go ahead. Sorry.
Oh, I was going to ask if usually this happens between the same species of plants, or does it actually connect? I mean, is it the same species or different species? Sorry, that's what I meant to ask.
Both.
Both. Okay, very good.
Yeah, both. And, you know, keep in mind that the plant here on the right has no incentive to help out the plant on the left necessarily, especially if it's a different species, right? But a lot of plants use the same. A lot of plants use the same kind of triggers to initiate defense, and so it just happens that one plant can help out the other one.
So is this considered altruism?
No. It's a good question. Altruism means that out of no benefit to yourself, you contribute something that damages yourself in order to help some other organism, right? So the plant here that's getting chewed on is already making these compounds. Okay. So it's not expending extra energy for that. They're just getting shuttled over to the next plant.
Does that make it like commensalism then? Or is it just kind of like this is happening and it's not an actual relationship, it's just kind of a side effect?
I think of it like a side effect. I don't know. It's kind of an existential question.
I mean, my idea is that the fungus manipulates the whole situation. And I mean, in their interest would be to protect all the plants that they're associated with, right? Because they get more food, more nutrients.
Yeah, they want the plants to survive because that's their sugar source.
Right. And so it seems like they would just, you know, send signals, whatever, to help every plant they're associated with survive, not out of the goodness of their heart, but only simply because they want to keep them alive so that they eat more.
Yeah, I mean, I think it's always safest to think of all biological relationships as essentially being like pretty cutthroat and amoral. I don't know that we've ever actually found examples of true altruism in biology. The closer we look, we start noticing that there is a benefit to the individual that's helping out. So yeah, I think I take kind of the pessimistic, this is the fungus kind of looking out for its own interest. It doesn't really care. The plant on the right doesn't really care about the plant on the left. It may get some benefit though, because if all the plants around it are super susceptible to the insects, there's going to be a higher pathogen load in that region. So by basically making everyone around you ready to fight off insects, that kind of keeps them away from everybody. This has led to some pretty out there kind of pseudoscience hypotheses, right? And this notion of the wood wide web. If you read any of Paul Stamets' work, he's a really good mycologist, really great at growing things and marketing himself as a great mycologist and everything. But he talks about like the power of moon energy and the forest's neural network of mycorrhizae. It gets a bit weird for me. Okay. It's also the inspiration behind that Avatar movie, right? Where everything's kind of connected through this root network. It's not exactly like that. So I think we already kind of touched on this question, what's in it for the fungus? Sugar. Sugar's in it for the fungus.
I have a quick question.
Yeah.
So, sorry, going back to the last slide, talking about how the mushroom kind of acts as that bridge between the two plants.
Mm-hmm.
What if let's say the tree was actually, let's say, an aspen tree where it's part of a grove and they're all interlinked?
Mm-hmm.
Can just a small group of mushrooms still act as a branch between let's say just the outer barrier of the grove and then still affect the entire grove? See what I'm saying?
Explain it to me again. I'm sorry.
All right, so looking at the slide, if the tree on the right was part of, let's say it was an aspen, which is part of a grove, and
it's all one individual. Yeah.
Would it, if that aspen tree has more shoots that produce more trees near it, can that mushroom still provide those mechanisms to the rest of the grove, or is it just that one tree?
Yeah, in that sense, it would be to the rest of the grove, because for aspens, an aspen grove is a single individual and they're all linked together.
Okay.
And so the fungus, by giving to one, would be giving to all, since it's a shared.
So if that one fungus was, could it travel through like the entire grove? As far, let's say the grove was a mile wide, it could still go that distance?
Yeah.
Wow, that's amazing.
Yeah.
Okay.
And, in fact, the largest organism on Earth is a honey fungus. It's not an ectomycorrhizal. It's actually parasitic, but it's bigger than the biggest aspen grove in terms of grams, because a single individual is almost 3 miles across in diameter. And it's linking together all these trees, and it's eating them all really slowly.
Wow, that's amazing.
Yeah, long distance transport can happen. Obviously, there's like a limit in terms of time and how long a hypha can be, but if they're all linked up together, little patches, and those are all linked, then yes, it's kind of getting to everything. Okay. So let's talk about some cheaters of this system. When I mean cheaters, what might I be referring to? There's always an incentive to cheat in a mutualistic relationship.
So, from what I remember, I don't know where I read it from somewhere, there's also fungi that pretend to help the plant so they can leach off some sugars, but they don't. But I thought they got kicked off by the plant, that it can somehow tell that they're leaching.
Yeah, those do tend to get kicked off. And that would be a cheating scenario where they're taking the sugars, not providing anything. But we wouldn't call that like a mutualism. That's a parasite. And so there have been several lineages of ectomycorrhizal fungi that have kind of shifted into parasitism, right? They just basically stopped sending the nitrogen, and they're keeping the sugar, and they're keeping the nitrogen for themselves. Now the plant isn't happy about that, because it's just something taking its sugar, okay? But it's still kind of helping the plant, because it's coating their roots, they're getting that pathogen protection, that kind of thing. And so in that sense, those fungi can be considered cheaters, in that they're not doing what they used to for the plant. They're still kind of helping, though. But there's even something a bit weirder because of these systems, and it's called mycoheterotrophy. These are achlorophyllous or achlorophyllous plants. Okay. So here we've got some plants that cheat the mycorrhizal network. What are they doing? Judging from their color. Guess what are they not doing?
They're not producing carbon.
That's right. So they don't have chlorophyll. Some of them have various degrees of chlorophyll, but there are quite a few plants out there that don't even make chlorophyll anymore and cannot do carbon fixation. So it's plants that can't turn air into sugar. So they're really bad at being plants, but they're still out there, still living, and this is how they do it. Okay. We've got our actual autotroph here, this plant, taking mineral carbon, making it into sugar, and sending it out to its mycorrhizal fungi. Okay. If those mycorrhizal fungi are also associating with a mycoheterotrophic plant, it's taking that sugar from the fungus that was taken from the plant. And so they're kind of just like dipping their fingers into this system and stealing carbon from this mycorrhizal network. They used to be able to make sugar. Like, I mean, look at these plants. You can't be a plant and be that color and have evolved unless somebody's given you some sugar. Okay, so their close relatives still make sugar out of air. A lot of these are like in the Vaccinium family, so they're blueberries, like closer related to blueberry plants. But this mycoheterotrophy is pretty odd. They've been associating with mycorrhizal fungi, and basically, if that mycorrhizal fungus is also getting sugar from another plant, these were allowed to acquire mutations in their rubisco pathways for fixing carbon, and then boom, they're done. You can find these here in Utah.
Do they provide, they must provide some benefit, though, the fungus. I mean, it's otherwise, why would the fungi associate with them? You know, fool me once, fool me twice. I mean, you know, it just seems like over generations when they just, there must be something that they're providing to the fungus.
Not necessarily. What does malaria provide to humans?
Well, good point. A lot of pain.
I think these are actually considered, to the best of my knowledge, like pure parasites on the fungus. They used to provide something to the fungus. Okay. But they stopped and the fungus hasn't really caught on.
I didn't figure it out. That's
hilarious. And keep in mind that that fungus is actually still dependent on getting sugars because it's lost a lot of its genes for making enzymes that break down complex carbohydrates. So it needs to be associated with the plant now to get its sugar. And so any of the pathways that it would use to kind of kick these plants off, kick off the good plant. Does that make sense?
That does make sense, yeah.
And so I feel like it's probably, they're kind of, to some extent, at least in any short term, like a million years or so, it's going to be pretty difficult for them to kind of like get rid of this parasite. Now, fortunately, these plants tend to be a lot more rare than the trees that they're associating with. How many of you have seen any of these up in the mountains here in Utah?
Probably the middle one.
Yeah.
Okay. Yeah, these are all, I put common in quotes, scare quotes for you because they're not actually super common, but you can find them. Okay. This is my favorite plant in the world right here, the Indian pipe. I just think it's so adorable. And it doesn't fix carbon at all anymore. It's totally just leaching off of this mycorrhizal fungus. Okay, so you'll only find these plants near the trees where these mycorrhizal fungi grow. Okay. So in this case, pine trees for these three. All right. So this is just to kind of show you how we track this stuff. This on the left is a figure from this paper right here, and this is all about plants. So this is from the Annals of Botany, and they're basically showing that you can grind these things up and throw them through a mass spec, and these plants look different than other plants. Okay, so here we've got autotrophic and circular, partially microheterotrophic. That means they are leaching off, but they can still make their own sugar. And then fully microheterotrophic, like the Indian pipe plant, is the dark squares here. Okay, so this was done in 2 different forests, and this is the ratio of N15 and C13. What do you see? What do isotopes like this actually, what can they help us learn?
How do you extract them, probably, right? Like with the source, the isotopes?
Yeah, the source. And so this is kind of the same thing. When you know, the carbon dioxide is rising exponentially around Earth, and there's an open question, or not actually open anymore, but there was, of, you know, where is it coming from? Is it that we've got, you know, just soils emitting more CO2? Is it from burning fossil fuels? Okay, how do you tell? Well, you take that CO2, you run it through a mass spec, and you look at the ratio of the different isotopes of carbon. And different plants, like different photosynthetic pathways, will preferentially fix different types of carbon. Okay. The carbon that we're seeing in our atmosphere right now looks exactly like the carbon that ferns fix. So it's got the same ratio of this heavy carbon that ferns do in their plant tissues. Why?
Because ferns fossilized a long time ago. They used to be dominant over the earth.
All the oil that goes into your car is fern carbon. Yep. And so that was carbon that was fixed a long time ago. So looking at isotopes can help us trace the origin of things. When we do this for humans in different cultures of the world, we can also see patterns. This is pretty interesting. Americans look like corn carbon. Yep. Asians look like rice carbon. Okay. So you can really kind of track people's or plants' diet. All right. So what we're seeing here is that the carbon and nitrogen signatures of these trophic plants are really different from all these other normal green plants. Okay. Because they're getting their carbon and nitrogen from fungus. Okay. And over here on the right is a really cool study where they used quantum dots. Has anyone heard of those? It's actually a group over in physics here at UVU studying quantum dots. So you may have seen posters in the hallway or something. It's like a little cadmium core with some coating on it, and you can conjugate it to some molecule of interest, like glycine, the amino acid. And it binds to it and it fluoresces just because of some quantum stuff, physics, blah, blah, blah. Okay, what's really cool about it though is that if you conjugate it to, say, glycine, you can track the flow of glycine through an entire ecosystem. Okay. We're still not allowed to go out and like pour these on the ground because they have cadmium and that might cause cancer or something. So we don't do that. But in the lab, we can set up experiments like this. Okay. Here we're looking at the flow of chitosan and glycine, and we're seeing it enter from the soil into the fungus and then up into the plant and even turning up in the leaves. And so we're getting much better at kind of tracing these mycorrhizal relationships and how nutrients flow through an ecosystem. And what we're finding is that in forest ecosystems, unlike grassland ones, the thing really regulating the flow of all these compounds, all the carbon, all the nitrogen, are ectomycorrhizal fungi. So even though they're only associating with maybe 2% of plant species, in a forest system, they're the traffic directors moving all the carbon and nitrogen through that entire system. So any questions about ectomycorrhizal fungi?
I guess earlier you mentioned how much of the plants depend on them, and you said 2%?
2% of the species of plants.
Of the species of plants. Got it. Okay. Sorry.
Yeah. I would think it's more like 90% of the biomass of plants. Given that, yeah, they're so common, like the northern boreal forests. Pretty much all conifers that we think of when we think of a woodland are dependent on ectomycorrhizal fungi. So you go up into the mountains here in Utah, up in the Uintas, and you're looking at an ectomycorrhizal system. It's all those conifers. Yeah.
Hey, Dr. Zong.
Yes.
Quick question. I remember at the beginning of the course we were talking about growing those mushrooms and how you wanted to get like a really robust protocol and how to be able to do those or whatnot. Did we have to swing past the biology office again to get the necessary stuff to do that, or do we already have it?
Oh, you've got it. I mean, we're talking like old newspaper, that kind of stuff. A bucket with some holes cut in it. Yeah, have you taken a look at the protocols I put up online? It's just like a link to YouTube series.
Oh, okay. So is that on the what is it called, the Hillbilly Mycology type stuff?
It's down at the bottom of that page.
To be honest, I didn't know we had that up, so I will look at it and let you know if I have any questions. Thank you.
Okay. Yeah, it's easier than you might think. The tricky part is keeping things clean. Back when I was first learning to grow mushrooms, I built myself like a little glove box with some old like dishwashing gloves, duct tape, and like clear giant Tupperware things. I would put my cultures in there with a bottle of Lysol, seal it up, stick my hands in the gloves, spray everything down with Lysol, and then open up and do my transfers. That's about how clean you have to be, which is pretty annoying, and so that's why it's like an optional assignment. But essentially, if you can go buy an oyster mushroom from the Asian market or go find one on a tree stump, you just rip it open, take it out, stick it on one of your agar dishes, and once it starts growing, you can use that to inoculate a bunch of pasteurized newspaper, and you'll get mushrooms.
And then they're edible and clean.
Yep. Mm-hmm.
So Dr. Zong,
yeah.
If you do it that way and just transfer it from your agar dish over onto the onto the newspapers, I know you're just barely describing why you have to be so clean, but at what point does that like ultra cleanliness come in on the transfer?
Yeah.
Because if we're not clean, what, it will just kill it off?
If you're not clean, other fungi that grow faster, Ascomycetes, will take over, and you just get this like green mold everywhere. Yeah.
Okay, got it. Thank you.
Yeah, that is the bane of every mushroom grower is the green mold. Yeah.
All right, I got a question.
Okay.
We probably covered it at some point in the class, but I don't remember it in how depth. Have we gone over fungi attacking other fungi? Because I've seen that on the wild ones I've collected.
No, we haven't. We haven't gone over that. I wasn't planning on it, but I can. So those are mycoparasites. And there are entire groups of fungi that specialize on eating other fungi. So I can throw together a little bit of material on that for you and stick it up on the web page.
Cool, thanks.
Mm-hmm. Yeah, there's this, I don't know that they grow in Utah actually, but one of my favorites, wild edibles, called the lobster mushroom. And they kind of taste like lobster. They're really good. But they always get attacked by mycoparasites because there's a group of fungi that specializes in eating nothing else but lobster mushrooms. And so I have this like really hateful relationship with mycoparasites. They run one of my favorite foods.
Maybe it's because it's so good.
It is so good. It's just so yummy. All right. Again, reminder, no class Thursday. Try to get caught up on homework assignments and such. And I will try to get caught up on grading. And then, okay, next week is lichens. And this 2016 paper that you'll be reading is a massive bombshell that like broke the world of mycology that year. We always thought that lichens were one ascomycete and one photosynthetic partner, but it turns out all of them have this like hidden cryptic thing, Basidiomycete yeast, in them that nobody knew about, even with all the DNA sequencing that we did. Okay, so they found some, and I remember being at the conference when this was announced, and it was like, what? A bunch of mushroom nerds falling on the floor in astonishment. It was pretty cool. So this is a nice paper, pretty short, but you get to see a lot about lichens and what goes into this crazy relationship. Because a lichen is an individual, but it's not one species. So it's pretty crazy.
Dr. Zahn, do we
Yeah.
have a quiz then on Thursday, still due on Thursday, or no, since there's no class?
No reading assignment, no quiz this week.
Beautiful, thank you.
Mm-hmm. All right, I'll check you guys next week.